Hello students, today we are going to discuss about the inhibitory genes. Inhibitory genes, it shows the ratio 13:3. It shows ratio 13:3. So we know that the epistasis means there are the two genes, one is the epistatic and another is the hypostatic. Epistatic and hypostatic. So the epistatic means which masks the effect of another gene, and hypostatic gene means it get masked its effect by the epistatic gene. So the inhibitory gene, it is the epistasis in which there are the epistatic and hypostatic genes. So for example, or assume that there are the two genes here, the one, suppose capital A, capital A. So A is the epistatic gene while B is the hypostatic gene. A is the epistatic gene and B is the hypostatic gene. Okay. So when the A, it may be in homozygous condition or it may be in the heterozygous condition. That means it may be the capital A or capital A, small a. It always expresses his character over the another gene, that is B. It may be in homozygous condition or heterozygous condition, or it may be in the recessive condition. Okay. So in such condition, the capital A, capital A, capital B, capital B, capital A, capital A, capital B, small b, then capital A, small a, capital B, capital B, capital A, small a, capital B, small b, or capital A, small b, small b. All these such conditions of the genotype, all they produce the similar type of the phenotype. Okay. And when this epistatic gene, it is in recessive condition, then and another, it may be homozygous or heterozygous, it produce the another or different phenotype. Okay. So here we will draw, here we will explain, take one example, that is, there are the two genes, a capital I, it is the epistatic gene, and capital C, it is the hypostatic gene. So when it is in dominant condition, that means it may be homozygous or heterozygous, means it may be capital I or capital I smaller. In that condition, the C, it may be in homozygous dominant, or it may be heterozygous, or it may be the recessive condition. It always masks the effect of C. Okay. So we will draw here one table. So epistatic gene, hypostatic gene, and here phenotype. Okay. So first, when epistatic gene, which is the epistatic gene? It is the I. Okay. So when I, it is in dominant or homozygous dominant condition, or it may be in dominant heterozygous condition, it always masks the effect of capital C, capital C, small c, or it may be small c, in that case, the C is responsible for development of the color and I is responsible to inhibit the color. I is dominant. So when I is in dominant condition, so whatever may be the C, it may be homozygous dominant or it may be the heterozygous dominant or it may be the recessive. It do not produce the color, so it produce white. Okay. The second condition, when it is in recessive condition, small I, and here capital C or capital C small c or small c. That means this hypostatic gene, it may be in homozygous condition or heterozygous condition or in recessive condition. So no recessive conditions, capital C or capital C small c, homozygous and heterozygous condition. It produce the color. It produce color. Okay. And the third condition, when both are in recessive condition, so again there is no development of the color and it again remain white. Okay. So the example, we will discuss about the example. So there are two different types of the fowls. Fowls, kumde. Fowls or the bird, a hen bird or the cock. So that is, one is leghorn. It is white in color. When it cross with Plymouth rock, it is also white. Okay. I will draw it again. Leghorn, which is white, when it cross with the Plymouth rock, it is also white. Okay. But genotypically, this leghorn white, it has the dominant condition of the genes capital I, capital C. Okay. And this Plymouth rock, it is in recessive conditions of small I, small c. Okay. So it is the P1 parents. It is genotype. It produces the gametes capital I, capital C. It produces the gametes small I, small c. Okay. When these are the gamete 1. When they cross with each other, they produce capital I, small I, capital C, small c. Capital I, small I, capital C, small c. Okay. So these are the F1s and they were white in color. Why they were white in color? Because this I, it is in dominant condition. So it masks the effect of C. So all F1 were white. Okay. So when this F1, they allow to self-fertilize, that means the selfing. Okay. So on selfing, they produce the 4 different gametes because they were heterozygous. So the first gene I, capital I. Then second small I. Then the second, that is capital C, small c. Okay. Similarly here, capital I, small I. Then second, that is capital C, small c. So in this way, these 4 gametes were produced. Okay. Now, when these gametes they put in the Punnett square. So in F2 generation, the 2 different types of the progenies were produced. So 9:3:3:1 ratio, it changes into 13:3. Okay. So let's draw the Punnett square. The 4 rows and 4 columns. Okay. So 1, 2, 3 and 4. 4 rows. 1, 2, 3 and 4. These are the 4 columns. So male gametes and female gametes. Okay. So capital I, capital C first, then capital I, small c second, small I, capital C third, small I, small c. Okay. Similarly, here also capital I, capital C, capital I, small c, small I, capital C, small I, small c. Okay. Now produce the organisms by crossing these gametes. So here the capital I, capital C, capital C. So C is responsible to develop the color. But I is the epistatic gene and it is the inhibitory gene. It inhibits the color formation. Okay. So when I is in dominant condition, it do not allow C to express its characters. So this fowl was white. Okay. Then this capital I, capital C, small c. Similarly this capital I, capital C, small c. Here also I is in dominant condition, so it do not allow C to express its characters. So it again white and white. Okay. Then this condition capital I, capital C, capital C. Similarly, this capital I, small I, capital C, capital C. Here also I is in dominant condition, so it produce white, white. Then this middle 4, capital I, capital C, small c, capital I, capital C, small c, capital I, capital C, small c, capital I, capital C, small c. Okay. So here also, in all these cases also, the I is in dominant condition. So it inhibits the color formation due to the C, and therefore it is also white, white, white and white. Then here capital I, small c, small c. Here capital I, small c, small c. Capital I, small c, small c. Here I is dominant. Therefore, it again white, white. Here I, capital white. Okay. Now these 3. So here the I is in recessive condition, C is in dominant condition. So small I, capital C, small c, small I, capital C, small c. So here I is in recessive condition and C it is in dominant condition. It may be homozygous or heterozygous. So it produces the colored fowl. Here colored, here also colored. Then the last one, small I, small c, small c. There is no I is dominant, no C is dominant. Therefore, it again white. Okay. So this 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 and 13. This 13 were white. It is 1, 2 and 3. 3 were colored. 3 were colored. So 13 white and 3 colored. Okay. So the Mendel's 9:3:3:1 ratio is completely get changed into 13:3. Okay. So in today's lecture, we have discussed about the inhibitory genes. The inhibitory genes means the epistatic genes. It inhibits the color formation or it inhibits the phenotypic expression of another gene. So I is inhibit the color formation, therefore it is the inhibitory gene. So when I is in dominant condition, I is the epistatic gene, so it masks the effect of C, that is another hypostatic gene which is responsible to produce the color, and therefore wherever the I is in dominant condition, so it inhibits the color formation. So the 13 they were white and 3 were colored. Okay, so the 9:3:3:1 ratio is get completely change into 13:3. So Mendel's ratio here completely changed. Okay, so it is known as the inhibitory genes or inhibitory gene epistasis, because here the I, it is the dominant gene which is responsible to inhibit the character of hypostatic gene. Therefore, it is known as the inhibitory gene epistasis. Okay, thank you very much. I hope you got the points.